It is my great pleasure to present a study conducted by Professor Wenjun and Professor Tao Lia's team from the Jinan University. The title of this study is The Mediating Roles of Gut Microbiota in the Associations of Air Pollution and Meteorological Factors with Metabolic Syndrome. With rapid economic development, dramatic changes in lifestyle, living environment, and dietary habits have contributed to a continuous rise in the global prevalence of metabolic syndrome, or METS. It is estimated that by 2035, the prevalence of METS may increase to about 53%, making it a major global public health challenge. In recent years, increasing evidence has suggested that environmental factors, including PM2.5, NO2, and ambient temperature, may play important roles in the development of METS. At the same time, the gut microbiota has attracted considerable attention as a key regulator of metabolic homeostasis. Previous studies have shown that environmental stressors may disrupt microbial composition and function, thereby promoting inflammation and metabolic disturbances related to METS. However, it remains unclear whether environmentally induced alterations in the gut microbiota contribute to the development of METS. Therefore, this study aimed to examine the associations of long-term exposure to PM2.5, NO2, and ambient temperature with METS, and further evaluate the mediating roles of the gut microbiota. This study has three main highlights. First, based on gut microbial composition, participants were classified into four distinct community clusters. Cluster 1 was characterized by the enrichment of Bacteroids, whereas Cluster 2 was characterized by the enrichment of Prevotella. Participants in these two clusters showed lower waist circumference, fasting blood glucose, triglycerides, and blood pressure levels, as well as higher high-density lipoprotein cholesterol levels. Second, we observed that higher exposure to PM2.5 and NO2, as well as lower ambient temperature, was associated with an increased risk of METS. Third, we found that key genera such as Megamonas and Megasphaera may play mediating roles in the pathway linking environmental factors to METS. We identified several gut microbial characteristics associated with METS. First, compared with non-METS participants, individuals with METS had lower gut microbial alpha diversity. Second, based on gut microbial community structure, participants were classified into four clusters, which differed in METS prevalence and dominant genera. Finally, compared with participants in clusters 4, those in clusters 1 and 2 had lower waist circumference, fasting blood glucose, triglycerides, systolic and diastolic blood pressure, as well as higher high-density lipoprotein cholesterol level. The covariation network showed that genera enriched in the non-METS group formed a relatively denser positively correlated module, mainly including Bacteroids, Faecalibacterium, and Parabacteroids. In contrast, genera enriched in the METS group were more dispersed across the network and did not form a similarly dense module. The univariate Permanova results showed that geographical location explained the largest proportion of variation in gut microbial community structure. Among non-geographical factors, NO2, PM2.5, and ambient temperature had relatively high explanatory power, suggesting that air pollution and meteorological conditions are important factors associated with gut microbiota composition. Finally, we further explored the mediating roles of gut microbiota in the associations between environmental factors and METS. First, we identified two contrasting association patterns between gut microbial genera and environmental exposures. Megamonas, Megasphaera, Menacoccus, and Bacteroids were positively associated with NO2 and PM2.5, inversely associated with ambient temperature, whereas Pseudomonas, Lactobacillus, and Acrobacterium showed the opposite pattern. Second, we found that higher exposure to PM2.5 and NO2 was associated with an increased risk of METS, whereas higher ambient temperature was associated with a lower risk of METS. Third, 6 gut microbial genera may mediate the associations between environmental factors and METS risk, with Megamonas and Megasphaera showing stronger mediating effects. In summary, our findings show that METS was associated with lower gut microbial alpha diversity, distinct community clusters, and differences in key genera, with enrichment of several potentially pathogenic genera. Long-term exposure to higher levels of PM2.5 and NO2, as well as lower ambient temperature, was associated with an increased risk of METS. In addition, genera such as Megamonas, Megasphaera, Menacoccus, and Ruminococcus may mediate the associations between environmental factors and METS risk, suggesting that the gut microbiota may represent an important biological pathway through which environmental factors influence METS.